Cool. Thanks a lot. Thanks for inviting me. I'm super happy to be here and have an opportunity to show my work. So what I'm going to be talking about today is from a previous postdoc at Uppsala University, and it's about non-ecological speciation and the genomic basis of it in a pair of sympatric songbird species. So, to give a bit of context, what I mean by speciation here is a gradual evolution of reproductive isolation. And reproductive isolation means a reduction of gene flow, which first of all can occur across individual in a landscape. So if we think about a population, for whatever reason, this gene flow between parts of this population can be interrupted, which can result in independent evolution and later speciation. However, this reduction of gene flow also occurs across loci in a genome, which means that specific loci within a genome might experience less gene flow than others. And this is because there is recombination, which create different types of allele combinations. And some of these combinations can survive better than others or be selected against all four, which means that specific combination might introgress and others might not. And this is interesting because it means that, first of all, the process of speciation can occur in the face of gene flow and is not necessarily interrupted by gene flow. And it also means that speciation can start at specific loci in the genome, and with extension of linkage disequilibrium, it might extend through, as shown here, to cover broader parts of the genome that don't introgress over time. When we speak about speciation, there are two main aspects that are very often put forward. One is the geographic context of speciation. So we often hear about allopatric speciation, sympatric speciation, and this basically defines how much gene flow can occur during speciation. And then there is also an ecological context, which I'm going to detail a bit more. Because it's quite important here to have an idea of what differs between non-ecological versus ecological speciation. So in non-ecological speciation, what happens is that we start with a population in a specific environment, adapted to its environment, and this population is divided, but the two populations remain in similar habitats, which means that in terms of ecology, they experience the same selective pressures. However, because they are independent, drift will lead to the fixation of different alleles, and these alleles might not work well together, which means that these different fixation can lead to incompatibilities. And if enough incompatibilities build up, then hybrids may have a lower fitness, and this creates reproductive isolation. In an ecological speciation model, we also start in a given environment, but what is important is that part of the population will evolve then in a distinct environment. So it might be, for example, colonization of a new place or change in habitat in part of the species range. And when population experience that, they will experience also local adaptation to reach a fitness optimum in their own habitat. And this adaptation might lead to different phenotypic adaptation, like big size in birds. And what is important is that this local adaptation can also lead to reproductive isolation. For a very simple case, for example, is that hybrids might have an intermediate phenotype which work in neither habitats, and they will be selected against. So this is another way to lead to reproductive isolation. Now, this is a very shallow introduction to speciation. There are many ways to evolve reproductive isolation, many types of traits that can be involved, but this is very general context. Another thing that is interesting to me is that the process of speciation doesn't really end with the evolution of reproductive isolation. And to see that, we can think about a concept called the speciation cycle, which explains how speciation events allow a buildup of biodiversity through time. So we again start with a population. This population experience speciation. The way speciation occurs is not important. What is important is that we end up with two species. And the key part here is that after speciation, the two species must be able to expand their range and typically evolve sympatry. And this is because speciation generally involves a reduction in range size. And if species would keep having a smaller range size, they would end up disappearing. So if we want to increase, like to have more species appearing than disappearing, this step of range expansion and evolution of sympatry is super important. And then it allows to restart the cycle and get more and more species. Where this is tricky is that evolving sympatry between closely related species is quite difficult because first of all, they need to avoid excluding each other competitively. So it usually means that they have little ecological niche overlap. And second of all, they need to be strongly isolated because otherwise they would create a hybrid swarm. So for this thing to work, it usually takes a lot of time, and species that are sympatric are typically species that have been diverging for a long time. In this talk, I will speak about all these processes using a genus of songbird as a system, which is called the wagtails, genus Motacilla. It's a fairly small radiation, and it's not at all a adaptive radiation. All these species have pretty much the same. They are very, very similar, except in plumage coloration. There is no divergence in typically ecologically associated traits like big shape or size or body size. And one thing that is quite fascinating is that there are two branches of the tree where plumage phenotypes evolve very, very fast, quite explosively. One is the white wagtail here, which is a single species with many different subspecies with lots of different plumages. And the other here is the yellow wagtails group, which has different lineages and again, a lot of different plumage phenotypes. So my talk will be focused on these guys. In this group, we can define three lineages that are based on molecular phylogenies are well distinct. The first one is the western yellow wagtail in green on the map, which has 10 subspecies that you can see here, which differ in terms of the coloration of males in breeding plumage. So basically, it's the same blueprint. It's a bird with a black back and yellow belly, but the coloration of the head changes a lot from all black to all white, some yellow, some gray. There is a lot of diversity. Then in pink, you have the eastern yellow wagtail, which is essentially the same thing, but it has three different phenotypes that are a bit different, and it doesn't occur sympatrically with the western yellow wagtail. They are parapatric. And finally, there is the citrine wagtail, which is here, and this one is very different in terms of plumage, very easy to identify with a gray to black back, some broad white on the coverts in the wings, and a completely yellow head. And this one is sympatric with both yellow wagtail lineages. Where this is interesting is that these species have very similar ecologies. They are very generalist, and sometimes citrine wagtails and yellow wagtails breed exactly at the same place, like a few meters apart, and they can use the same habitats. If we gather all this information in terms of phenotypes and geographic distribution, and we try to think about how these species should be reproductively isolated, or all these lineages, sorry, it seems that both Eastern and Western yellow wagtail lineages have a low isolation because overall their phenotypic differentiation is low. As I said, they are parapatric, and in the area where they meet, which is mostly here in Siberia, phenotypes actually integrate very gradually. So they seem to interbreed a lot in this area. On the other hand, the citrine wagtail is very easy to diagnose in terms of phenotype, very different. It is sympatric with both yellow wagtails over very broad areas. It's not like narrow hybrid zones, it's really broad areas of sympatry. And in these places, phenotypes are very consistent. There is no sign that they hybridize a lot. And also hybridization is very rarely observed in nature. So it seems that this species is strongly isolated from yellow wagtails. So in terms of representing that as a phylogeny, keeping in mind what I said about how long it takes to become sympatric with a closely related species, what we expected when we started working on this system is that yellow wagtails, both lineages, are recently diverged. They didn't accumulate a lot of differences. And the citrine wagtail is more deeply diverged, and it had time to accumulate differences and go back into sympatry with yellow wagtails. So to get to the bottom of that, I'm going to present results which are all based on analysis of whole genome resequencing data of 112 wagtails, which cover all the range of the species. You can see them as the markers over there. And yeah, when we look at the population structure of this group, so here shown as a PCA based on genome-wide SNPs, we have a picture that is fairly similar to the phylogeny I showed earlier, where the citrine wagtail is well differentiated. There are no intermediates that could indicate recent hybrids. The two yellow wagtails are also quite clearly differentiated. However, there are some individuals in the middle that are likely admixed. And these correspond to the birds from Siberia where there is phenotypic intergradation. And what's important to take into account here is that east and west of these birds, we don't have any samples. We have huge gaps. So it is likely that if we had a better sampling, we would find like a gradient of genomic transition here rather than discrete clusters. However, when things get interesting is when we build a phylogeny based on this data. And what we find is that the citrine wagtail here is sister to the western yellow wagtail, with the eastern yellow wagtail sitting outside. And we again have these presumed hybrids from Siberia, which are intermediate, so that makes sense. However, it's super surprising that the citrine wagtail is nested within the yellow wagtails. And this could mean two things. One could be that recent processes, post-divergence processes such as introgression, could blur the phylogenetic signal and make the citrine wagtail look more closely related to the western yellow wagtail. Or it could mean that the citrine wagtail is a lineage of yellow wagtail that evolved a different plumage at a faster rate for some reason. So one way to gain insight into that is to look how phylogenetic signal changes across the genome. So to do that, we cut the genome into smaller windows. We calculate a phylogenies into each window, and then we look at how support for different topologies are located in the genome. In this case, it's a fairly simple topology with three tips. So we have three possible trees, which are here. And when we do that for all the autosomes in the genome, we find this distribution. So there is no clearly one topology that is much more common than the others, which indicates probably a lot of incomplete lineage sorting. However, we find this topology, the first one, where the Western yellow wagtail and citrine wagtail are sisters, is the most common. And even more surprising, the third one, where the yellow wagtails are monophyletic with respect to citrine wagtail, is much rarer. It's really uncommon. Okay, so as I said, this is a fairly simple phylogenetic problem. In a free taxa topology, if we assume that there has not been ancestral gene flow, the most common topology should agree with the species tree. However, there are several things that go against this interpretation. The first one is that the distribution of the two discordant topologies, so the two less common, is asymmetric. If the variation would only come from incomplete lineage sorting, we would expect them to be in the same proportions, these two. So this supports that there has been introgression. Furthermore, there is an association between the topology that is supported by a genomic region and the recombination rate of this genomic region. So genomic regions with a higher recombination rate, shown in pale gray, tend to support more the first topology. And when you go to genomic regions with a lower recombination rate, you find more support for the alternative topologies. This is important because we expect to find less introgression in regions of low recombination. And this is because typically there is selection against introgression, and regions of low recombination purge this introgression more efficiently. So yeah, there has been gene flow, and this makes knowing the relationship of the species quite complicated. So at this step, it's very tricky to know whether the citrine wagtail is an unusual yellow wagtail that evolved a very different plumage, or if it's just introgression that mixes the phylogenetic signal. One way to get insights into this question is to understand better how the genome themselves evolve and what region of the genome may specifically give us better phylogenetic signal. So a tool that we have for that is what we call studying a genomic landscape, which is calculating specific statistics in windows along chromosomes and then looking at how they vary. And here specifically, I'm looking at differentiation, or FST, which measures how, on a pairwise basis, how how alleles fix differentiately in two populations. So the higher the FST, the more alleles are different. On average, when we compare the citrine wagtail to either yellow wagtails, we find an FST of 0.19, which compared to other songbird species pairs is not very low. It's fairly expected for a closely related species. However, we find regions where it's much more elevated, which we call differentiation islands. So this one is very striking, for example. So these islands could indicate processes such as selection acting specifically in the citrine wagtail genome, and that make it more differentiated from the yellow whitetail. However, if we take the same scan for comparing the two yellow whitetails, which is here, we see that the background level of differentiation is much smaller, twice as small, which means that they are more recently diverged or have more gene flow. But these big islands where FST is high, we still recover them. So this one is most striking, but we have others which are shared. So these are shared islands of differentiation, and it's interesting to understand where they come from. So these shared islands of differentiation are characterized by a higher FST compared to the rest of the genome. This is how I defined them originally, so it makes sense. And surprisingly, they are also characterized by a lower absolute divergence, or DXY, which measures the number of mutations that accumulate after the split. So we have different alleles that fix, but we don't have an increase of mutation, an accumulation of mutations. And furthermore, we have in these regions a decreased nucleotide diversity. So within each species, there is less diversity of mutations. And what's even more interesting for me is that if we include outgroups, so comparisons with outgroup for FST and DXY, and nucleotide diversity in outgroups, we also find the same pattern. So what does that mean? This can be interpreted as a strong signature of background selection in these regions, which is that selection purges deleterious mutations. And this also decreased diversity in neutral variants that are under linkage disequilibrium with these deleterious mutations. And in birds, the recombination landscape is very conserved, which means that background selection can act repeatedly in the same region for a very long time scale and deplete the diversity in these regions over long times. And the fact that this is shared with outgroups tell us that this action of background selection has been going on for longer than the divergence of yellow and citrine whitetails, and even longer than the divergence between these and other whitetail species. And this is interesting for us for phylogenetic inference. First of all, because if these genetic sequences have been under selection for a long time, it's unlikely that it's less likely that they have been affected by more recent processes after the split of yellow and citrine whitetails. But more specifically, because of this selection, effective population size is reduced locally, which reduces the likelihood of ILS, incomplete lineage sorting. And they are also located in regions of the genome that is of extremely low recombination rate, which reduces the likelihood of finding introgression, as I explained earlier. So if we look at the phylogeny in these regions, what do we find? As a reminder, this is the support for free topologies in all the autosomes. But if we consider only these shared islands of differentiation, we find this pattern, which is very different. And here we have free topologies in equal proportion, 1/3 each, and with very similar distributions. More concretely, what it means is that in a free taxa phylogeny, having the free possible topology in the same proportions is expected if the divergence time is extremely short with respect to ancestral effective population size. So basically, it means that this branch must have a much bigger NE than length. And this idea is supported by a polytomy test, which rejected the hypothesis of a bifurcation at the root of the tree. So if we look at that in a more visual way, we expected a phylogeny like that, with a long internal branch reflecting variation in phenotypes and reproductive isolation, but actually it seems to have happened like that with a pretty much simultaneous split of the three lineages. And we've tried to date this split. It's not easy because of the gene flow, but it seems to have occurred between 500,000 to 1 million years ago. Okay, so with that in mind, one question that's really puzzling for me is why did, if the species tree looks like this, why did reproductive isolation and plumage differences evolve faster in citrine wagtail compared to between the two yellow wagtails? One way to get at this issue is to look for signals of positive selection that are specific to the citrine wagtail, that could tell us that positive selection have acted at this specific loci and favored a particular evolutionary outcome. So here is another chromosome with a FST scan, and here I've highlighted one peak that is found in the first two scans when the citrine wagtail is involved, but that we don't recover when we compare only the two yellow wagtails. However, one issue with this approach is that all regions of elevated FST are located in regions of low recombination, where we expect that differentiation will accumulate faster regardless of selective processes. So it makes it really difficult to untangle these things linked to recombination and clear signal of positive selection. To try to get a bit further with this, what I've done is to use a topology-based approach. So I've split the genome in tiny genomic windows of 20 SNPs. For each of them, I have calculated a tree, and then I've searched for windows where citrine wagtail haplotypes are monophyletic. So the genomic background looks like this. There are a lot of allele sharing, so you see that the citrine wagtail in orange are a bit everywhere in the tree. There is no clear structure. However, what I'm looking for is something like this, where all the citrine wagtails are along the same branch, together monophyletic. To be clear, this is not an explicit test for selection because we expect that lineage sorting occurs through neutral processes within the citrine wagtail, regardless of selection. However, given the presence of introgression and very high level of allele sharing across all lineages, selective sweeps are the process that is most likely to accelerate lineage sorting compared to neutral processes. So when running this analysis on autosomes, I will not talk about sex chromosomes today because it's really complicated. But on autosomes, I've found 513 windows where citrine wagtails are monophyletic, which cover 700,000 base pairs. It's a very, very tiny proportion of the genome. A bird genome is approximately 1.2 gigabases, so this is really small. And these regions overlap 60 genes. However, of these 60 genes, we have 67% of them that also overlap the shared islands of differentiation that I talked about earlier. So haplotype sorting in these regions is not linked to citrine-specific sweeps, but just background selection and neutral processes. So this means that regions where there is this strong signal of sweeps are very, very localized in the genome and don't concern many genes. Most of the remaining windows where there seems to be a citrine-specific pattern going on are overlapping two genes. One is on chromosome 1. It's HNG box transcription factor, or BBX, and its function in birds is not well defined, so I'm not going to talk a lot about it. but another one is on chromosome 20, and it's called agouti signaling protein, or AZIP. And this one is really interesting because it's involved in melanogenesis. In lots of organisms, it separates pale versus dark morphs or species. So if we look at what the uh, genetic landscape looks around this region. So in gray here in both graphs, you have the coding region of the gene AZIP. And this graph here shows with the black dots how monophyletic the citrine whitetail is. So when it gets to 1, it means that it's fully monophyletic. So this is monophyletic, and you see that around it there is a lot of allele sharing. In these regions, we also have a peak of FST, which is shown here with the solid lines. And we have shown in this graph a reduction of the nucleotide diversity in the citrine whitetail that is not found in the yellow whitetails. So background selection, for example, would reduce nucleotide diversity in all species if it occurred in all species. So we would not expect to have a peak here. But the fact that it's only in the citrine whitetail is more consistent with a selective sweep. So this signal of selective sweep is welcome and interesting because, as I said, this is a gene that is involved in melanogenesis. And if you look at the citrine and yellow whitetail, you can see that one way they differ a lot is with the mantle coloration, which is dark gray to black in citrine whitetail versus green in yellow whitetail. And this means that the yellow whitetail has less melanin deposited in the mantle. And also more white could mean that there is no melanin deposited. So clearly changes in the AZIP gene could underlie these differences. What's interesting, however, is that this doesn't explain the wool variation in plumage. So why don't we find other color-related genes in this analysis? If we relax a bit our criteria and we don't select windows without allele sharing, but windows with extremely low levels of allele sharing, what we find is that there are two other color-related genes that match this criteria. First one is KIT ligand, which is also in the melanogenesis pathway. And the second one is beta-carotene oxygenase 2, or BCO2, which breaks down melanins and favors the expression of yellow colorations. So this is relevant for the head coloration because yellow whitetails have melanin deposited there, and citrine whitetail don't. They are fully yellow. If we look at the same graphs as for AZIP in terms of differentiation, nucleotide diversity, etc., we see a very similar patterns. We have increase of FST in this region. We have a decrease of nucleotide diversity only in Citriola. So it seems like there has been a selective sweep there. However, why did we not find it in the phylogenetic analysis? Why is there still haplotype sharing there? A way to get deeper into that is to actually look at the nucleotide sequence in this region. So as an example, here I show the phylogeny for a small region close to BCO2, where the citrine whitetail, as you can see here, are monophyletic, right? If we look at the nucleotide sequence in this region, we see that all citrine whitetails have the same haplotype fixed. There is no variation there. So this is very strong signal of selective sweep. However, we also find that two yellow whitetails are heterozygous for this citrine whitetail haplotype. So they carry it on one chromosome, and on the other chromosome, they have a different haplotype. And if we look at the rest of yellow whitetails, so here there's a small sample, we see that there is a lot of variation. So a specific haplotype has fixed in the citrine whitetail, but it's still also existing in yellow whitetails at the heterozygous state. And largely, if we look at all SNPs around in this region around BCO2, so in the broader region, including the BCO2 gene, we found that 84% of them they are fixed in the citrine, but they are shared with other species, including outgroups. So this means that most of this variation is ancestral standing variation that is like transient through the whitetail radiation and might be also reshuffled with introgression. This is difficult to separate both processes. Okay, so I'm going to now wrap all that up. So when it comes to the phylogenetic pattern, the divergence pattern of these three lineages, to me, it's very consistent with what we call budding speciation, which is the idea that within a large population, a given group of individual evolves, becomes reproductively isolated from the source population, but the rest continue to extend genes. So in that sense, phylogenetic relationships don't reflect how reproductive isolation evolved. And in this specific case, it's quite interesting because the divergence is not extremely recent, and still this phylogenetic pattern has been conserved. One way to get there is if a population is isolated at the periphery of the range, it's called peripatric speciation. So the idea is that you have a large ancestral population which splits at some point. So in our case, it's a three-way split of ancestor of Western yellow whitetail, ancestor of Eastern yellow whitetail, and ancestor of citrine whitetail. And what is important is that one of the population gets isolated at the periphery and is much smaller than the others. After that, we have phenotypic divergence, and we might have selection on other types of barriers that we couldn't characterize. And what we see is that the Eastern and Western yellow whitetail didn't diverge too much, but the citrine whitetail, which is a much smaller population, diverged a lot and evolved a completely new phenotype. And this was subsequently followed by range expansion, and this new phenotype allows the citrine whitetail to cohabit in sympatry with the yellow whitetails. And this part, in this case, is quite remarkable because, as I say, evolution of sympatry is a slow process, and in birds, it's usually at least 3 million years, typically more 4 to 5 million years. So this is very fast. Under a model of peripatric speciation, we can expect two things. The first one is that there would be asymmetry in range size, which is true at speciation. However, in our case, we cannot really test that because there has been range expansion since then. So we don't know what the geographic ranges looked when speciation occurred. And another would be a demographic bottleneck in the citrine whitetail. Because if it got isolated in a small peripatric population, we would expect that its effective population size decreased. And it's something we find when we reconstruct the history of effective population size. So after the split of the three lineages, the citrine whitetail has a bottleneck, while the yellow whitetails has population expansion. However, okay, this has to be a bit relativized because usually peripatric speciation is proposed when the peripheral populations are really isolated and small. And here the smallest effective population size is 200,000 individuals, which is not small in the absolute. It's just smaller than the yellow whitetails. And also the genome-wide nucleotide diversity is um, is still quite large in the citrine whitetail compared to many other whitetail species. So it's unclear to what extent this peripatric speciation could explain how the phenotypes evolved. We've also learned with it a bit about how plumage evolves in this system. We have found that positive selection in the citrine whitetail at three, at least, genes could be linked to plumage differences. Two of them are in the melanogenesis pathway, so linked to melanin synthesis and deposition. And another one is linked to melanin breakdown and the expression of yellow colorations. Furthermore, the strongest signals of sweeps were always found outside of genes, and we couldn't find any missense mutations. So this supports that regulatory regions are more important. So it's more a question of gene expression than real changes in the protein sequence. And this is something that is very often found in birds, so it makes sense. And finally, we found that a fixation of standing variation in the citrine whitetail drove the evolution of these regions. So if we come back to the question that I said initially, which is why did reproductive isolation and plumage evolve faster in the citrine whitetail? One thing that we can say with good certainty is that positive selection on standing variation is associated with the plumage differences. It's really difficult to imagine or even more to know how originally this new phenotype originated, and especially how it could rise in frequency to become dominant in the population. However, one thing we can say is that the emergence of a new phenotype likely facilitated reproductive isolation in sympatry because the two yellow whitetails have very similar phenotypes and are able to hybridize, while the citrine whitetail doesn't and could evolve sympatry. And the role of plumage coloration for prezygotic isolation in birds, especially songbirds, is quite important. So it would make sense that this is an important driver of isolation. And finally, there might be a role for demographic processes because yellow versus citrine whitetails have quite different demographic trajectories, but it's really difficult to test for that and give a more precise hypothesis on that. And with that, I would like to thank you all for listening to me and again for inviting me. And I would like to thank the team that works on that project with me, which is led by Per Ahlström at Uppsala University and also involved Leif Andersson, Ximena Caballero, Eric Embody, and Daria Shipilina. Thank you.